Okay, thank you so much. I really appreciate this great opportunity from ILSI North American for me to present this novel research design in this research design challenge. So as we know that efficacy tests normally do not design or power for the safety measurement. So our research aimed to develop a novel research design to detect the safety level of the bioactive soybean isoflavones in the efficacy test to determine the safety level of this compound using in the breast cancer patients. So we have invented a very new and novel soybean isoflavone-enhanced breast cancer therapy. So our study shows that the soybean isoflavone are able to resensitize the endocrine treatment, for example, tamoxifen in triple-negative breast cancer. So triple-negative breast cancer is considered very aggressive and lack of therapeutic options. So our study is very novel and important by providing the therapeutic approach for these patients. So we have done a lot of work on animals, and we would like to moving forward to push our study in the phase 1 clinical trial. So soy isoflavone, as a very commonly consumed diet nutrition, has received a lot of controversial debating. So major concern is the soybean isoflavone are considered as a phytoestrogen that may mimic the estrogenic effects and interfere some hormone pathway in the target organs. So there's still no direct evidence showing that high level of isoflavones is associated with the harmful side effects in human study. So if we want to move forward our study in clinical trial, since the major safety design problems or hurdle we face too is how to design or determine an effective safety study to identify the potential of very rare side effects and in human studies, in the efficacy human studies, when we use a very small number of humans in the phase 1 clinical trials, and when we use a very high level of isoflavone dose and then the recommended level. So to face this challenge, we have designed a very novel we have provided very novel research design in our study. First, we want to stratify the patients to lower and higher isoflavone sensitivity group based on their ability to metabolize or utilize the isoflavone in their systems. First, we want to test the S-equal as a major bioactive metabolite of the isoflavone in the patient urines. So second, we want to determine the gut microbiome that specifically contribute to metabolize isoflavone in the patient's stool samples. The third, we're going to test the epigenetic modulator change in patient blood. So you can see, using these 3 novel biomarkers, we are able to effectively stratify patients into lower and higher sensitivity patient groups based on their ability to metabolize isoflavone. Second, we would like to treat the patient with either lower or higher level of soybean isoflavone plus the endocrine treatment, tamoxifen, for 3 months treatment. So the differences with the lower and higher group is the lower group patients will receive the safety level of the recommended isoflavone, but the higher dose group patients will receive much higher, about 13-fold higher of the recommended level of the isoflavone. So you can see in each group, the patient has been labeled and separated for the high, depend their ability to metabolize isoflavones. So we are, by this setting up, we are able to effectively prioritize the patient with either efficacy or toxicity observation in these two patient populations. If we did not find any adverse effects in the higher dose group, we're going to move forward for long-term high-dose group observation to determine the potential long-term side effects in those patients. Third, we're going to observe the target soy isoflavone safety test in these patients. We're not just doing the conventional safety test. For example, the regular physiological parameters. We also wanted to do to test the specific side effects or adverse effects that are related to the soybean isoflavone, for example, the estrogenic side effects, and the combination side effects when we use these two compounds together. So in summary, there's three unique design elements in our study. First, we stratified the patient into lower and higher isoflavone sensitivity group based on their ability to effectively metabolize or utilize the isoflavones. The second, we can apply the different dose of isoflavone from a lower to higher, and also different time frame to from short to long-term time frame to prioritize either the efficacy and the safety test in these patients. Third, we can observe the target safety test to collect sufficient safety data in these patients. There's a lot of novel elements in our studies, not mention the component I mentioned before. Our study can also provide sufficient data to establish a safety intake level for future nutrition studies. And more important, our study developed a personalized therapeutic strategy in the triple-negative breast cancer patients who may not have enough or too much therapeutic opportunity. So our research team is composed by multidisciplinary experts, including cancer experts, physicians, toxicologists, microbiologists, and statisticians. So finally, I want to thank you for all the grant agency and IELTS North American giving me this great opportunity. Thank you.
And that was perfect timing. The timer just went off.
Thank you.
All right, we have time for questions from our judges.
I'll just leave it here. That three-way test or for potential interactions or potential sensitivities, have you validated that in other patients before? Has this been used in other settings?
We validate into our preclinical studies. We haven't done in the patients. We have done and published a lot of paper for this three-way test in animal study in the mouse, but haven't done any work. We just wanted to do a kind of design this study. Want to do in the patients. Okay.
I think I noticed when you were talking about the mouse study, you had some kind of X times higher that the mice have been fed. Could you tell us a bit more about that? Was it 13 or what was the
dose?
The
okay.
dose, yeah.
So we used the higher dose of isoflavone when we treat the patients. That's what I said. Why we have to face the safety problems if we want to do the clinical trials. We use 25 micrograms per kilo of the isoflavone when we treat the mouse, which is considered very high. So the recommended isoflavone is about 30 to 100 micrograms per day. So we use about 2,000 microgram per day in mouse. So it's about 13-fold higher. So that's why I said safety concern. And no one have found that high level can cause the harmful or adverse effects in humans. So we wanted to design this safety test to see whether there's a potential or side effects if we use a higher level of isoflavone in human patients.
Do you have a feeling already that the low dose might not be enough to help?
it could be, but it could be the low dose may not have it effective. But when we stratify the patient, divide the patient with higher and the lower sensitivity group, it's possible the higher sensitivity patients may respond to the treatment, but the lower may not be respond. So that is why we stratify the patients. So the patient will has a different, patient group has a different ability or possibility to develop either the efficacy test or the efficacy effective response or the adverse effects. So that is why we want to stratify the patients.
And my final question before I turn it over, have these women already been treated with some pretty severe treatments, and are they facing like there's not much else to do, and now you're thinking of this?
So a triple-negative breast cancer patient is normally do not respond to the normal therapeutic treatment. So that is why we want to develop the alternative treatment, chemotherapy for these patients. So if we want to do this clinical trials, the patients could already receive extensive treatment before they may not respond to the common or conventional chemotherapies. So that's the consumptions.
Thank you. It's a great presentation.
Thank you.
And an interesting concept. So I'm wondering if you were to recommend this for a different component, a different bioactive, how would you recommend that they do this? Like if you don't have that information for the 3 tests or 3 ways to do this.
So if we want to do the different components, first we wanted to inform what is the safety level for the regular human beings. What is safety level? So when we do this safety level, if we understand this is a safety range for using this compound, we can enhance or increase the level for example, 10-fold or 20-folders to see whether there's a potential toxicity using in the animals. If the animal didn't show any, the side effects, we can move forward into the human studies. So always we have to do a lot of preclinical works in animals in order to translate this type of study into humans. So.
Thank you. One more round of applause, please.
Thank you.